My talk will be only partially about F-sharp, but I use F-sharp to develop a lot of machine learning algorithms. But this talk is more sort of about computational biology and cancer research, how I use machine learning and other quantitative methods in cancer research. I am not a biologist, so if any of you has a biological background, please be patient. But I'll be speaking, I will give you some background on the biology, and then I will basically give an overview of a few algorithms, and then there will be some F-sharp at the end as well. So, what's cancer research about actually? Well, cancer itself is basically a disease of the genome. It happens whenever some base pairs in your genome get mutated and it doesn't get repaired. And just to give you some idea of the scale of the problem, this is actually in Cambridge. This is a photo from Cambridge where I work. This is a psychopath that leads from Cambridge to another neighboring village, and it's 1.5 miles long. And it's because it goes through the biomedical campus. They wanted to do something nice with it, so they painted these little stripes on the whole path. And this actually represents one gene, a human gene called BRCA2, which is quite an important gene that basically drives the genome. I will talk about it a bit more later. And it has 10,000 base pairs. So this covers the whole 1.5 mile path in Cambridge. So just to give you some scale, I think every talk that mentions DNA has to give some kind of comparison. So if we wanted to do a path like this for the whole human genome, we would have to go around the whole Earth 20 times. That's quite big, isn't it? So cancer can happen if some of the genes get mutated. And again, another point to give you some overview of the scale. The first human genome that was sequenced in 2003, and it cost $100 million. And it's been getting cheaper and cheaper. This is actually an old graph. Now we are around $1,000 per genome. And then sequencing of the whole human genome, 3 billion base pairs. And now it's getting to a stage where if you have some kind of problem, you will go to the doctor and they will actually take a sample of your blood or of your tissue and send it off to sequencing. So we are getting more and more data, and it's been projected that in a couple of years, genomics will have more data than astronomy, than particle physics, than YouTube. And YouTube has 300 hours of videos uploaded every minute. So that's already quite a lot, but genomics and this kind of research will have more data. So we are basically the ultimate big data science. Anyway, another bit of biology. This is the DNA, and the standard structure of the DNA is double helix. And that's the sort of stable representation of the code that encodes everything in our cells. And when cells want to produce a protein to react to something in the environment, for example, it sort of unzips in the location, and then there come other proteins that bind to it and that allow it to be transcribed into RNA. This is this molecule that's being basically copied out of the DNA. And that gets basically transported from the nucleus of the cell into some other parts where it gets translated into proteins. And this is very useful for research, because then we can basically look at how much of the RNA is in the cell for each gene. And that gives us an idea of what's happening there at the moment. So during some biological process, we can always basically extract the RNA from a cell and see what genes are being transcribed at any specific point. And that's very useful to observe what's actually happening there. And yeah, I already mentioned that cancer is basically a disease of the genome. usually happens when some genes are mutated. You can be born with some kinds of mutations. You can obtain them during your life through some environmental damage or basically by pure chance. And my lab is doing a lot of research on the BRCA2 gene, and these two genes act as basically chromosome guardians. There are two types of genes that are involved in cancer. One type is oncogenes, which are genes that when they get mutated, they cause cancer directly. And another type are tumor suppressors, which guard against cancer, and when they get mutated, they basically can't prevent cancer anymore. So these two genes are actually tumor suppressors, and they are heavily involved in, for example, some types of breast cancer. I guess you have heard about, for example, Angelina Jolie, who had a double mastectomy because she had the mutated variant of these two genes. If you have mutations in these two genes, you have about 75% of getting cancer, and it increases probability of breast cancer in men as well, and probability of prostate cancer. So it really involves everyone. But one of the main things is that cancer is not just a single disease. It's very, very heterogeneous because it depends on what mutations in which gene you get in the beginning. And generally, for example, when you get mutations in these two genes, they stop guarding the genome against other mutations. And then the mutations just accumulate, and there are more of them, and then the more mutations you have, the harder it is actually to cure the cancer you have. So this was a bit of an introduction into the whole thing, cancer. And as I mentioned, cancer is not a single gene. So how do we actually find out what type of cancer people have? Because even if you have cancer in one type of tissue, for example, lung cancer or breast cancer, it can have multiple different variants that behave differently in terms of treatment, that give you completely different prospects. So one thing that's used a lot in machine learning, and I was actually developing some algorithms for that during my PhD, is clustering. So in like normal setting, in machine learning, clustering can be used, for example, to cluster customers. This is an example of a simple small data set. It contained 440 wholesale customers, and those customers were restaurants, cafes, hotels in Portugal, I think. And the data set contains their spending on fresh produced milk products, paper, etc. And if we cluster this, we can get something like this. This is projection from the 6-dimensional space of their spending into 2D using principal component analysis. And what's nice, there are two big clusters, and the red cluster actually represents hotels that have much larger spending on detergents and paper than other customers. So this gives us quite a nice interpretation of the data. And we can use something very similar to cancer. If we map customers onto genes, and instead of their spending on products, we can look at their gene expression. And well, why would we even want to cluster the cancer data? Right now, if you go to a doctor and you get diagnosed with some disease, you get some kind of treatment. That's the standard way of treating patients in most settings. But people are not the same, and cancers are not the same. So what we really want is to basically distinguish patients into groups that behave differently in terms of different treatments, or that have different types of cancer. And this is an area called precision medicine. It used to be called personalized medicine, but that's not a very good term for it, because you won't get your own personal medicine, because that's basically almost impossible to develop, because all medicines have to go through some clinical trials. So now we switched the term into precision medicine. So this is actually a graph from my PhD thesis, where I developed some algorithms to cluster cancer data. And this is data on breast cancer. It's a projection of 700 gene expressions into 2D. And each of these three groups has different probabilities of survival. So in the future, probably when you get diagnosed with cancer, I hope you won't, but if you get diagnosed, then the doctors might take a sample and basically classify you into some subgroup that behaves differently and give you targeted treatment. So once we basically create these subgroups of patients, what can we do later? How can we find out how they are different? So actually what I was doing in my PhD was designing algorithms to cluster multiple different data sets in a unified setting. Because the plot I was showing before was only using gene expression data. This is just graphics showing that I was using different types of data that describe different types of the processes that are happening in the cell. And because the cancer behaves differently in each of the different contexts, what we really want is to have some good clustering algorithms. I won't go into too much detail about that. If you want to hear more, I'll be at the pub, happy to discuss. So, once we actually put together some subtypes of cancer, we might want to see what characterizes the different subtypes. And for that, we can use another very typical machine learning algorithm called collaborative filtering. So, just to give you some idea how this works, a very successful application of collaborative filtering was in the Netflix prize. And the Netflix prize happened when before there were any machine learning competitions like Kaggle. I think the original one started in 2006, when Netflix offered a prize of $1 million to anyone who can improve their prediction algorithm by 10%. It got eventually awarded in 2009. So it took quite a long time. But the winning algorithm was using partially collaborative filtering. So how does that work, actually? The problem setting is that you have some customers and you have movies, and each customer rates some of the movies. And the problem is that you might have thousands, millions of customers, and you can have hundreds of thousands of movies. So this matrix is very, very sparse, and you want to fill in the missing data. And the simple algorithm called collaborative filtering uses matrix factorization, where this is the Netflix matrix, let's say. So it's very big and very sparse, and we can actually factorize it into a product of two smaller matrices, where one represents users. Each row in this small matrix represents a user, and it has the same number of rows as the rating matrix. And the second matrix represents movies, where every column represents one movie. And if we multiply them together, we get this full matrix, giving us ratings for movies that people didn't actually evaluate. And again, we can use a very similar algorithm in cancer research, where we map users and the patients and the ratings into mutations that the patients have in their genome. And one of the problems in cancer research is that most of the mutations appear only in a minority of people. Now that's quite a complicated thing. Most mutations, there are some mutations that are very, very common, and they appear across many patients, but there are many more very rare mutations that don't appear so often. So if you look at the movie scenario, it's something like that everyone sees a couple of very popular movies, but the rest is sort of like some people see it, some people don't. So how do we map that onto the Netflix scenario? Now here we have the patient, and here we have mutations. C mutates to A, etc. And This big matrix decomposes into a product of two smaller matrices again, when one represents the patients, basically gives us summarized characteristics of each patient, and the second matrix represents, they call it mutational signatures of cancer, where each mutation has some kind of characteristics with respect to different types of cancer. And for example, substitution of C to T and vice versa is very typical in smokers. So this is correlated with environmental damage. And we don't really know why this happens. This is part of the research that was published actually 2 years ago, and the research is still ongoing. So we don't know why that happens, but this kind of substitution is very typical for tobacco users. And in practice, we don't have to use just one letter substitutions. We can look at larger sections of the genome. So this is a very nice machine learning algorithm that gives quite an interesting insight into what actually happens in the genome. And now I will move into a slightly different area of research that's not really about machine learning, but it's still using computational methods. And this is actually a research of my colleague Van Hall, who's been looking at using computational methods for drug design, for example. Because now I was showing you how we can actually discover different subtypes of cancer, but what can we do later? We really want to design some drugs, right? So this is the part that will actually contain some F-sharp in the end. And for proving that, well, what we want to do when we are designing a drug is that we want to basically destabilize the cancer cell, to force it to self-destruct, which is something that should happen normally, but in cancer, this mechanism is broken. Normally, when a cell starts dividing without any bounds, it gets self-destructed because there are control mechanisms in the cell. And so we are trying to get some kind of target for a drug that will destabilize the cancer. And what we can use are formal verification methods, because we can basically specify a biological system using simple rules and then test what will destabilize it. So I'll just give you some sort of little introduction into the methods that we are using for that. So you have probably seen some SAT problems. If we want to solve this kind of formula where A and B are Boolean variables, we can just run SAT solver, or in this case, you can just look at it. And this is satisfiable if A is true and B is true. And this is very simple. Well, it's hard to actually solve it in a general setting. But what we are using to model the biological systems is actually something called SMT. It's now not SAT, it's SMT, and it stands for, I'll first explain what it does. Basically, here we have just some variables that had Boolean values. And we can replace these variables with predicates that have the Boolean value when they are evaluated. So for example, here we have A. Here it would be true or false, but we can replace it with A is larger than 3. And now when we evaluate this part, it will be either true or false. So this is a simple modification. There can be any type of predicate, and it's called the satisfiability modular theories, because you can use predicates from any type of theory. And now this is satisfiable for A equal 4 and B equal 0. So this is a way how to get integers or even real numbers into the standard satisfiability setting. And I will show you some example in F-sharp, because what my colleagues are using is the Z3 theorem prover. If you haven't seen it, this is actually an atom. This is an atom with the ionized plugin. which you can use to run any F# code. And I just loaded the Z3 theorem prover. Can you see it? Okay. And what I will do here is I'm creating a context for the SMT solver. And I will try to solve a simple problem where, given a price, I want to see the minimal number of banknotes that I have to pay the price. So in this case, I just have $1 note and $5 note. So I'll just create an integer in my context, one with value 1 and another with value 5. And what I created here is an arithmetic expression that takes the $1 note and multiplies it by one multiplier, and $5 note that multiplies it by another multiplier. And now this sums the two together. This is just an arithmetic expression. And for any number of M1 and M5, it will give me if it's true or false if I add some kind of target value. So let's say we want to pay $42 with 10 bills. And I will create a solver and add some kind of constraints. Namely, a constraint that the total value that I can pay with my banknotes will be equal to the number of to what I want to pay, and the total number of notes will be equal to 10. And now this is ugly, but I can run the solver, and it will tell me that it's satisfiable if I use 2 $1 notes and 8 $5 notes. So that's quite nice. And I can actually make it even a bit nicer, because this was just computing it for a specific number of nodes. And I actually rewrote the parser a bit using quotations. So now I can put in the expression using this kind of notation, where I have two variables, A1 and A2, and A1 times 5 plus A2 times 1 equals 42, which is the target value. And now I can have a simple recursive function to test every number of nodes that I want to use for the payment and find the smallest one. So each iteration of this function creates the solver, adds the conditions, and tests if it's satisfiable. So I will just evaluate that. And we will start with 1. And actually what I'm getting is that it's unsatisfiable for 1 banknote, but it's satisfiable for 2 if I pay 10 $1 bills and minus 8 $5 bills. That's not right. So we actually need to add some added conditions. Just copy that over. Okay. So now I've added a condition that says for every variable, make it greater or equal to 0. And let's run that again. And now it found a solution. I need 10 banknotes. And yeah, I turned the timing off. Let's turn it on again. And let's try it for something a bit more complicated. Let's say we have 3 different banknotes. The other one will be, let's say, $10 one. A3 times 10. And it will be equal to, for example, 3,542. And you'll see how quickly that runs. And I think most of the time is spent printing. And it took 3 seconds. So I need two $2 bills and 354 $10 ones. That's not very surprising, but I hope it gave you some kind of idea how this type of solver works. So what it is actually used for in normal setting is for software verification, where you encode some preconditions and postconditions and loop conditions and assertions in these types of formulas, and then you run the solver and it will tell you if your program is satisfiable or not. For example, I think Airbus uses a variant of this solver to verify their implementations of their flight programs. This is an example from ChIP-seq, which is Z3 applied to C# programs, where we can have some kind of function and add conditions. So this function tries to swap two items in an array. So what we want to have is that the first index is larger or equal to 0 and smaller than the length of the array, and the same for the second one. And now if you want to run this function and you are using ChIP-seq, you basically have to prove that these conditions hold. You can't run it otherwise. And if these conditions work, then you can safely assume that the rest of the function will work fine. So how can we actually use it in the biological system setting? Well, we can again map proteins and genes and receptors in cells into variables. And because we are looking at quite a dynamical system, for each time point, we basically want something like this to hold. For each variable, that means for each gene, let's say, if the value of the gene is equal to its target value, then we'll keep it the same. And if it's smaller than the target value, we will increase the gene expression. And if it's smaller, if it's larger than its target value, we will decrease the expression. And we can define the state of the biological system using these formulas. And we can run the solver, and that will give us the next state. And we can iterate this until the system stabilizes. So my colleagues actually implemented this in a model called BMA, Biomodel Analyzer. And I have some example here. Okay, so this is a model of 5 skin cells. And what happens in skin is that the bottom layers of your skin are constantly renewing and growing, and the top layers of your skin are basically dying and falling off. And we need to get a balance in this, because if your skin would be growing faster than it would die off, then you would get skin cancer. And if your skin was dying more than it was growing again, then that's not cancer, but it's really bad as well. So, so we want this system to stabilize. And they basically looked at literature, published models for skin renewal, and put this model together. And they even extended it into a 2D model, and they found bug in the existing models. Some variable that all existing models weren't counting for. And
so our skin has a bug in it?
Well, the model didn't stabilize. The model wasn't stable. But we can call it a bug because they used software verification tools to find that. Anyway, they used it in cancer research as well. And they used it for one type of cancer called chronic myeloid leukemia, which is very simple from a system point of view. So this is a normal situation where you have chromosome 9 and chromosome 22. And what happens in this type of leukemia is that these two chromosomes cross over and they exchange their parts. So what happened here is that the part of chromosome 22 got bound to chromosome 9 and vice versa. And unfortunately, this happens in a place where there is one gene here, and they basically exchange parts and create a mutated gene that causes this type of leukemia. So it's very nice in a system point of view because it has very clear starting point. And this is a model they came up with. They collected a lot of information from literature about known interactions between proteins and genes and receptors. And they came up with a model for this type of leukemia. And they actually found that this disease is incredibly stable. If you were using a drug that would target just one gene or one receptor, the system remains stable and the cancer remains in place. So this actually correlates with the clinical observations. And what we have to do in this type of cancer is to target at least 2 genes simultaneously. So that's a type of combined therapy. And they were actually able to identify the interactions that could be disrupted and that are disrupted through already existing therapies. And they also identified several new ones that can be potential drug targets. So this is quite an interesting application of software method applied to biological systems. The standard way how to model these things usually is through systems of differential equations, where the value of one gene depends on another gene, etc. But the problem is that these models don't scale very well. But using this type of prover, they can actually use local stability properties to infer global stability. So this allows applications on hundreds of different genes. So basically the most important part here is how to collect the information from literature, and that's still not automated. So I hope this gave you some kind of idea about using computational methods and machine learning methods in cancer research. So yeah, machine learning is not just for advertising or trading. If you are interested in some of the details, here are some links. The slides are already online, and there are some links to papers that describe the applications. And if you have any other questions, here is how to contact me.
What we're using for like a wrapper around Z3, like I didn't see it fully back there, but like with the quotation, is that just all online? You're doing it there, or is there some sort of library you're using?
The wrapper around Z3, they have the .NET wrapper.
I mean, like for you like decompose the.
Oh yeah. Yeah, here I created a quotation, but what I was actually using is this wrapper. It uses pattern matching.
Okay.
Basically, what it does here is that it first decomposes the quotation into the variables and into functions. Those quotations basically give you the evaluation tree of an expression. And here I'm basically just matching variable is a variable. Yeah. If I'm calling a multiplication, then it will call the multiplication in that tree, etc. And I can use this to compose a whole expression.
Is it online somewhere?
Yes, it is online. It's on my GitHub.
So, to what extent is this technique applicable to, like, I guess, all cancer types? I guess really my question is, like, are there any cancer types to which this type of analysis is not well suited, or yeah?
Well, the main limiting factor is the availability of existing information on existing interactions. Because if the type of cancer is not very well studied, then it's very hard to develop a model like this.
When you were talking about the collaborative transfer, you had this matrix factorization. Which factorization was that?
Well, what they actually used for the cancer data was just a non-negative matrix factorization. Because you can impose different kinds of constraints in collaborative filtering. And generally, if you make it non-negative, it's more interpretable.
What's next for your research?
Well, for me, I'm just finishing my PhD and moving on to a postdoc, and I'll be working on pancreatic cancer, and I have quite a lot of data about basically different types of the systems. A lot of different types of genomic data and gene expression data. And my task is basically to get as much information out of that as possible. So I will probably be using a lot of machine learning methods to extract interactions. Yes.
What's the computational complexity of that satisfiability problem you're solving here? Is it NP-complete or even worse?
In general, it's NP-complete. Because it basically reduces it to a sub-solver, and that's NP-complete.
Right. So when you're dealing with large amounts of data, what are the limitations? I mean, at what point does it become infeasible to solve in cancer research specifically?
Well, this type of solver is actually quite clever because it doesn't solve the whole system at once. It solves it locally. It solves the satisfiability in local modules, which are then composed together.
So it's more like a greedy approach.
Yes.
Cool.
Well, it's a bit more clever than that.
So,
But, well, the models I was showing, actually, these ones that are online, you can basically run it, and now it's trying to stabilize the system, and it said it stabilizes, and it says 0 seconds.
Mm-hmm.
So,
Cool.
this type of model, which has about 100 variables, is quite easy. And here you can also look at how it did the stabilization and what are the values of the individual variables.
So how does the research determine the cancer behavior? Do you say, oh, now I'm going to throw a wrench in it and take this out, and then see if it stabilizes, and if it doesn't, then it's a probable expression of cancer, and if it does, then it's a resilient system? Or?
Well, in this cancer example, if the model stabilizes, that means the cancer process is not disrupted. But you can... yes.
You're trying to disrupt the cancer process
Yes.
with the treatment.
Yes. But there are, the treatment is represented by disabling certain interactions or disabling certain proteins in the cell. But this type of model was used even to basically tune or adjust existing models for other types of biological systems. Because if the model stabilizes, then it's likely that it's representing the true underlying process, that the researchers didn't miss any crucial part of the system.
Is there something special about this Z3 solver? Like, I mean, SMT solvers have been around for a really long time. I'm just wondering, why do they choose to use this one?
Well, from what I've heard, maybe Thomas has a bit more to say about that because he did programming language theory and things like that. But it's the most widely used one.
Okay.
It's the most practically usable one.
What kind of solver is this for? Like a constraints programming, linear programming,
It's
mathematical
equivalent to a constraint satisfaction problem. So online you can find some examples using Z3 solver to solve Sudoku, for example. So it's an equivalent problem to constraint satisfaction.
So I would imagine you'd have to model it particularly if you're going to be using, like, I guess, Boolean things to kind of, so then it would, when switching from one state to another, it's like always going to switch. Whereas if it were like partial differential equations, then it would be modeled as just kind of along a continuum, right?
Well, that's why this is using the SMT solver. It doesn't have to be just Boolean.
Okay.
It can contain real values, but in this biological example, they generally set the values to either Boolean values or 3 values. I mean, for example, for a gene that's downregulated, that's neutral, and upregulated. So it's a simplification. but in general, the systems based on differential equations, they can handle about 10 variables.
So they basically model it basically on high, medium, low, or something.
Yeah, yeah. It's a simplification, but it works quite well. Actually.
How widely is this technique used? I mean, is this like the first time that this kind of take on this problem? Is this the first time that this technique has been used, applied to cancer research, or have there been like previous attempts, using similar techniques?
Well, most previous attempts were using the differential equation approach, because that's a bit more natural. But it's relatively new. It's been developed over the past 5 years, basically. And the application on the leukemia data set, that was actually published this year.
What we actually can see on this chart, because I'm trying to figure out what is the difference between every circles. Does it show something particular, or just is like a notion of cells? What is inside? Because they look like almost the same.
Well, they are the same basically, but it's a layer of skin cells, and the cells are the same.
So
they just need to stabilize in the sense that they need to be created in one end and die in the other. But the cells themselves are the
so
same.
what you're saying, it is dying one of this piece and need to be replaced by another one, or some part of this piece is dying?
No, they're going off in layers.
Yeah.
And in a sense, is that what you're showing is that each skin cell, once it's formed, was identical to the one that occurred below. The only problem is when you start to get changes in any one of those, such that either they die off quick or they
grow
too quickly.
Then it can create cancer. But the cells themselves are identical.
I mean, part of the problem with skin cancer is that skin cancer is driven by UV mutations, which are a function of how much UV light can actually penetrate your skin. So if you don't have a lot of dead dead skin cells at you form a barrier, you're going to have a higher rate of UV mutation, which will probably affect some of either whatever the left or the right side, which is supposed to be the inside, is going to change the mutation rate, which can probably throw the system out of balance. I assume that's what's going on.
Yeah. Well, this wasn't actually about modeling specifically cancer. It was just about modeling skin cells. So this wasn't the cancer example. But yeah, it's the right intuition.
But it sounds like you can have both. You can have both occurring, in the sense that you have mutations occurring from below a layer that could even be exposed, and then you can have additional mutation from exposure from above too.
Because there are many different mechanisms how mutations can occur. The UV light can cause mutations. Exposure to radioactivity can cause mutations.
You have a base rate of mutations which occur just a replication.
Right, but you can have something that can throw out the base rate.
This doesn't model mutation.
But this is just a standard model of skin. This was meant just as an example of a biological system.
You'd mentioned before the skin dies faster than it grows below, that you've got a problem. Do you know if that's the definition of necrotizing fasciitis? Skin flesh-eating disease?
I suppose you can say that as well. I'm not a biologist and I'm not a medical professional, so I am just a mathematician. So I think you would have to speak to someone else.
You mentioned at the beginning of the presentation that the cost of sequencing has gone down dramatically over the last 20 years. What's the driving force behind it? Is it just the cost of hardware going down, or is it also the availability of computing resources like cloud computing or frameworks for parallel data processing, things like that?
I will just jump to that slide.
Yeah.
Yeah, you can see that here it was just falling down quite gradually,
right.
And here we basically changed methodologies. So now it's something called high throughput sequencing, which is better than the system that was used before. Before it was so-called Sanger sequencing, where they used very laborious methods to actually extract each base pair. Now we have something slightly better, and there are new methodologies coming as well, which will probably allow even people who don't know anything about the machines and anything to sequence genome just on their bench, basically. So it's change of technique, change of methodology.
In the sequencing, do you know if there's a big data cleaning problem?
Well, one part of the problem is that the sequencing data are incredibly noisy. And it's also quite complicated to get the actual genome, or when you are looking at gene expression, to actually find out how much of each gene is being expressed in a cell. Because the standard way to do sequencing, the mainstream one, is that you basically split the genome into smaller segments, and you read each of them. And they are only about 150 base pairs long, typically, and you have to assemble them. And typically you have a reference genome that you are trying to map these reads onto, but it's an incredibly noisy process. So that's also a big challenge right now in genomics, how to make this more reliable. But hopefully the methods are getting better and they are getting more precise. So I believe in a few years this won't be such a big problem.
I saw on the slide, or rather the website with the blood stem cells, that was on Microsoft Research. What level of stewardship or oversight or involvement did Microsoft have that project? Just out of curiosity.
Well, that project was originally developed in Microsoft Research in Cambridge, but it's in collaboration with other research institutions. But my colleague who was heavily involved in that is actually not in Microsoft Research. He's in the same department as I am, which is the MRC Cancer Unit, and we are basically completely independent. So it was mainly because the people who were working on that were in Microsoft Research at the time it was developed. But Microsoft Research has quite a good department doing computational biology.
Do you know if they use F-sharp?
Yes, they do. Actually, the whole backend behind this is written in F-sharp.
I think one of the guys came to compose last year.
Yeah.
I thought the video should be online.
Yeah, it's Sami.
Ishta.
Yeah, he was involved in this project as well, and he's still in Microsoft Research. All right. Are there any other questions? If not, then thank you.